on this computer. Let's try that. Okay, so the test will be set up the same basic way. And I'm going to share my iPad screen so I can write. Hopefully I can do this. Good notes. Share screen, iPad, share, install. Okay, now we gotta do this. Wait, and my iPad's going slow. Oh, I know. Hold, this is frustrating. That's not gonna work because the regular computer, will be. Oh, let me try. I'm afraid if I do this, I'm gonna lose you guys. Hold on a minute. I had to change my settings. And watch me like disconnect all of you. I hope not. Network. If I change it to Wi-Fi. Oh, watch this poop out. If you go away. Connect. All right, now let's try this. Yeah. Okay, so same number of questions. Okay, so there, hold on, I'm gonna get you a blank screen. I'm so visual. I need to show you.
Dr. Blank, I put a question in the chat and I see that there's a couple of us that are agreeing with what I asked. So could you clarify that?
So it's going to be the same kind of setup. So the study guides are a guide. So let's look at some of the study guide stuff. I do pull the questions from the test bank. So if you go into the book website, there's a, have you practiced with any of the book stuff? No, maybe.
For myself, I've reviewed a lot of the study guides that we had.
Okay, so the study guides are good. So if you want, what you can do is, if you are writing out the study guides, if you email them to me, I will, I can go over them and see if you need to put in more detail or if you're missing something. Does that work? Like you could even take a picture of them and like do the phone scan thing and then just email them to me and I'll comment on them and give you some feedback.
Okay, thank you.
Yeah, I mean, that's one thing we can do. I made those practice questions, that little kind of study guide. You want to go over that and then I can fill notes in and you guys can ask questions from there?
Sure.
Okay, so it's basically the same. I mean, the test is going to be the same kind of idea. All my tests are kind of the same, where it's, you know, like 40 or 50 multiple choice questions and then some short answers at the end. So yeah, Melissa, the learning objectives are the study guide. So let me go to you guys can see on my weird apps. Let me go to D2L. Yeah, we can go over the practice questions. Yeah, and that'll help with the study guide too. So let me get to D2L on here. Maybe, come on, D2L. See if I can get in. Come on, log in. My browser's retro. Okay, so when you guys go, I'm just going to open up the class that's totally online, but the the 40 section, the one that's a hybrid, it should look the same. If it doesn't, you guys send me email so I can fix it. So if we go to content, okay, and then you guys go to what week are we in? 8. Okay, so if you go to week 8, and then under here, there's some, oh shoot, that's weird. It's rolling slow. Okay, so there's learning objectives for exam 2, right? So that's basically the study guide. So I had an old one up there. I took that one down before spring break. Sorry, but here you guys go. So here's chapters 7, 8, and then the PCR stuff from chapter 9. And then 13 is viruses. And then 14 and 15 is the immune system. So probably maybe the hardest stuff is going to be maybe the virus stuff for you guys. I'm not sure what'll be worse. So why don't we, where do you want to go through let's go through the study guide, and then well, not the study guide, I mean the practice questions. I was going to make you guys a key for it, or somebody give me feedback. What do you want? Do you want me to go over the practice questions and then I can fill in holes that way? Let's do that, I guess. Oh, somebody said study guide. There's a lot on the study guide. Okay, so if we start here, let me download it, and then I can put it in my thing I can write with my pencil on, hopefully. Download, yep. Okay, so here, new port. Oh, shoot. Right, now we're looking for the one that's today. Learning up there we go. But what? Okay, now you guys can see this. Okay, so first thing on here, can I upload? I'll try to upload it after I write on it. Yeah, yes. Okay, so if we go here, the first thing says, how's the structure of prokaryotic DNA different from eukaryotic DNA? Okay, so remember, first thing you have to remember is prokaryotes, that's bacteria. Okay, so prokaryotes are bacteria, so don't get that confused. And then eukaryotes are pretty much everything else. So like we're a eukaryote. We also have like plants or other animals or fungi or algae. Okay, so the first thing to remember is prokaryotic DNA, they only have a single circular chromosome. So there are some weird exceptions, but for the most part, prokaryotic DNA, it's just a single circular chromosome, right? They don't have, their chromosomes don't look like this, okay? Like you guys are used to seeing them. Eukaryotes look like that. Okay, so eukaryotic chromosomes, they're going to be linear. They're going to be in a nucleus. Okay, usually they're paired, right? So there's 2 alleles compared to prokaryotic. They only have 1 copy of each gene. Okay, now the nucleoid is the region in bacteria where we find the chromosome. Okay, so that's where the chromosome is. They don't have a nucleus. Okay, so that's going to be a big deal. So no nucleus, right? But they have a region where the chromosome hangs out. Okay, but it doesn't have a membrane, so no membrane-bound organelles. Okay, now transcription and translation and replication. So that's on those practice questions. So let me, I didn't really give myself enough room to write on here. Let's put a piece of paper up. Okay, so if we go, and this is something you learn in general biology. So when we do go from DNA to DNA. Okay, so if we copy DNA, that process is called replication. Okay, so replication just means we're copying the DNA. So bacteria do it, humans do it, everybody does it. In order to grow or repair any of that, you have to copy the DNA. Okay, so that's called replication. And that kind of makes sense. So you're just replicating it. You're doing exactly the same thing, hopefully. You want it to be the same. Sometimes it's not the same. You might have a mutation. So if it's not the same, there can be a change in the DNA. So in the sequence of the DNA. So instead of being A, T, C, C, G, A, or something like that, you could have something get messed up and the C gets turned into a G. Okay, so that counts as a mutation. Okay, so a mutation is a change that happens. Sometimes they're bad, sometimes they're not. Okay, so a mutation could cause a change and make somebody sick and cause a genetic disorder, or it could be beneficial. So like they keep talking about, like coronavirus is mutating, it changes. Okay, so if it's bad for it, we probably don't see it because the virus dies out or can't succeed. But if it's good for it, good for the virus, not for us, then we see those mutations, right? Because it makes the virus more survivable or better pathogen. Okay, so I kind of got off on a little squirrely thing there. Okay, now, when we go from DNA to RNA, okay, and lots of times we just say messenger RNA, this is called transcription. Okay, so we take admit. Okay, so we take the we take the DNA and we copy it and we turn it into RNA. And then in eukaryotic cells, we let the RNA out of the nucleus, right? So we let it out. So then the last step is called translation. And translation is how we actually make the protein. Okay, so my trick for telling people to remember this is, okay, DNA to RNA, the C is transcription, the C comes before the L. So in alphabetical order, this one comes first. So you have to go from DNA to RNA and then RNA to protein to actually make it happen. Does that make sense? Okay, now, so that's, let me go back to the, oh, shoot. Back to, how can I get back? Well, I feel silly. I don't know how to get back to my other screen. There. Okay. So transcription is we're going from RNA to DNA, right? And then, or no, I did it backwards, you guys. DNA to RNA. Sorry, I'm thinking about, is somebody chatting? Is somebody in the waiting room? How do those like video game streamers do all this? Okay, so DNA to RNA, and then from RNA to protein, that's translation. Okay, now if there's a mutation and there's some sort of change in the A's, T's, C's, and G's, if something happens and this changes, that can change the outcome. So that can change what the protein is. And it could be a good mutation, could be a bad mutation. Okay, now, what's a genome? Okay, so the genome is all of the genetic information in something. Okay, so it's the genome. Let me make this. So it's just all of the genes in an organism. Okay, and you guys have probably heard of the Human Genome Project. So I think that was in the, I'm trying to think, I can't remember the exact date, right? But the Human Genome Project, that was the idea that they were going to take a person's DNA, right? And they were going to sequence it. They were going to figure out all of the A's, T's, C's, and G's in it and try to figure out like what they meant and like what they meant. So they were trying to figure out, okay, if this gene is for this, or they were just trying to figure it all out. Like, what does it mean? Okay. And it actually, there's a ton of information about it, right? So it's kind of like too much information. So then they had to figure out, okay, they could do they looked at RNA, they've looked at the proteins that people have. So there's all these different levels where they're doing like these ohm things. And one of the ohm things is the microbiome, right? So the microbiome is another example. So it's just like all of the organisms associated with somebody's body. I was listening to some goofy, shoot, it was Mysterious Universe or something like that. And they were talking about how people have like electrical currents and they were calling it the electrome, which I don't know that that's a scientific thing, right? But it's supposed to make you guys laugh, but I can't see you or see if you're laughing. But okay, now a plasmid and a chromosome. Okay, so chromosome is all of the genetic, right? They were talking about whether like people were like glowing and how like to heal somebody, like what they could do for with their energy. And they were talking about everybody's electrome is different. I don't know, right? Okay, now what's the difference between a plasmid and a chromosome? Okay, so a plasmid is just, it's a circular piece of DNA and it's extra chromosomal. Okay, so the organism doesn't have to have it to survive. Okay, so it's not necessary for it to survive. They naturally occur in bacteria. Okay, so that's where they originally found them. So they're like what you can find them in the wild, right? So they naturally occur in bacteria. Circular piece of DNA, extra chromosomal, they're double-stranded, right? And they carry different genetic information, right? So the one that you guys know about is like pGlow, right? So pGlow is a plasmid that people made, right? And it's a circular piece of DNA. It's got the green fluorescent protein in it. So they took a gene from the jellyfish and they stuck it in a plasmid and they got it to work and got all sorts of things to light up with it. Okay, the other things that plasmids have is usually they have a resistance gene, so like that ampicillin resistance gene. Okay, so plasmids aren't essential. Okay, but if you took a chromosome out of bacteria or you took some person's chromosome away, right, it has some bad effect, it does something, hurts them, because you need that information to survive. Okay, so they're useful to scientists because we use them to make genetically engineered organisms. That's how they move DNA around. Okay, now a resistance plasmid is something that they're actually naturally occurring. That's where they originally found them. And they're important because they can, they say confer or like give antibiotic resistance to an organism. Okay, so that's actually bad, right? If it's happening in the wild. So if you think about somebody that, that's weird because what you guys are seeing does not look like what I have on my screen. Because I have to add another empty page. And I have to remember how I did it. Okay. All right, so antibiotic resistance genes. Or plasmids, okay? So say this is this is E. coli, that's normal, and it's just hanging out. Actually, probably the best one to do is say this is Staphylococcus aureus. Okay, so Staph aureus, it's normal. You find it on your skin, right? Everybody probably has it. So it's hanging out on your skin, and there is a way that it can pick up an antibiotic resistance gene on a plasmid. Okay, so what that's going to do is that can confer antibiotic resistance to the Staph aureus. So that is kind of one way that we got MRSA. So MRSA is methicillin resistance or resistant Staph aureus, right? So the antibiotic resistance gene got from one place to another, okay? And those antibiotic resistance plasmids can go from one organism to another, okay? So they can move around and they can go to another organism. And that's why we're supposed to take all of our antibiotic. So we'll talk about that some more, but they're really important because medically they're a problem because they create antibiotic resistance to organisms. So then the antibiotic doesn't work anymore. Okay, now a transposon. All right, so transposons are just ways to move a big piece of DNA. And I'm not too hung up or worried about that, okay? But it's a way to just move a chunk of DNA. And it's interesting because of one of the first geneticists that was female is the person that discovered them. Her name was Barbara McClintock. But she didn't get a real lab, like they gave her a closet kind of thing. But she figured out these, they call them jumping genes. So that they're genes that can move around. It's just, they're interesting, but you guys don't have to spend a lot of time on it. Okay, now a mutation, I already kind of talked about, right? So mutation is just how the DNA accidentally gets changed. Okay, so we have it on here. So something happens and the sequence of A's, T's, C's, and G's gets messed up. Okay, so it gets changed. So how can they be formed? Okay, so mutations can be spontaneous. Okay, so it's just an accident that happens when we're going through replication or transcription. Okay, so spontaneous, it's an accident. So something happens when your cell is doing replication or transcription, and it makes a mistake, right? So just like I make a mistake when I'm writing something or I misspeak or something like that, the same thing can happen in your body and with your DNA or in bacteria DNA or virus DNA. Okay, now, usually our body is really good at getting rid of these spontaneous mutations. So our immune system gets rid of them, wipes them out, and you never see them. So you only see them if they are able to survive. Okay, now mutagens are these other things that can cause mutations to occur. So they're going to increase the likelihood of a mistake. Okay, so examples of mutagens I have organized here. So chemicals, and then things like radiation. Okay, so chemicals are things like they go from like, I have them listed on here, like some that are not super bad and some of them that are really, really bad. Okay, so there are things that are, they call them a base analog. So a base analog is basically a chemical that looks like an A, T, C, or G. Okay, so I always think about it as posers. So it's going to come in and it's going to pretend that it's a T. All right, but it's really this T that's different, right? Some sort of like funky T. Okay, so if it comes in and it looks like it, but it's not it, it messes up the rest of this. Okay, so the DNA can't get read. Does that make sense? Yeah, maybe. No. Okay, now chelating agents, one example of that is mustard gas, right? And I don't know if you guys have ever, you probably have never done this, but like I've known people that have accidentally, like if you mix ammonia and bleach, don't ever do this, right? But that creates mustard gas, and mustard gas was used, I think, in World War II. I'm not sure that it was used in World War I, but it causes cancer. It's eventually, it takes away your breath. You can't breathe, and then it over the long run, it messes up your DNA. I guess I just kind of want you to know, there are different, like, no, there are different chemicals that exist that can cause a mutation. And like if you ever work in a lab, you're going to see, or like really anywhere, you see like symbols on different chemicals, right? It might say that it's a mutagen. So things that deaminate, like nitrates and nitrites. So I don't know if you've ever been pregnant, there's all these things that they say, oh, don't do this, because we don't want that to change the DNA of the baby, or of the cells that are going to become the baby if it's really, really early, right? So like caffeine, right? Nitrates and nitrites are found in like lunch meat and sausage and hot dogs and stuff like that. Acridine is one that they have used it to treat malaria, and it gets in, and your DNA, that's supposed to be a picture of double-stranded DNA. And like your A's are pairing with your T's and your C's are pairing with your G's, right? So what happens with these things is they get in here and they like go in between the pieces of DNA, and the DNA can't get read. So they're bad. And you see them a lot if you work in a lab, you run into stuff that's really bad. Okay, now radiation, these are things that you guys will be familiar with, right? So like UV light, what that does is it takes, if you have like T's paired with A's like this, what UV light does is it actually will mutate it, and it'll make the T's pair with each other. So then they're not working the way they're supposed to. They're paired like this, and they call them like thymine dimers. But it messes up the DNA can't get read. Okay, so same thing with like X-rays. There's just a whole bunch of energy coming in, and it damages the DNA, so it can't be read. Gamma rays, but like if you think about skin cancer, right? And if you give work with somebody, or you give X-rays, right? If you're in the dentist's office, they're monitoring how many X-rays, how much radiation you're exposed to, so that they can kind of control how much you're exposed to and that you don't damage your DNA. There are different kinds of mutations. So there are point mutations. So a point just means that like an A, T, C, or G is changed. So it's very specific. Frame shift mutation means that when we read DNA, we read it in like threes. So each word that we're going to read to be an amino acid, the words are 3 letters long, so 3 nucleotides long. So if something gets deleted, so let's say I delete this accidentally, something happens and I lose that. Okay, that is going to shift the word basically. So instead of being ATC, it'll make it ATG. So that'll can give me a new amino acid, and that means that my protein's going to be different. So it might not work the way it's supposed to. So it's almost like you can't read the words right, the way that they're supposed to be read. Okay, and then the other one I have on here is this transposon thing, which I mentioned earlier. And then this question, I think you guys can answer because I just talked about that, right? Oops, sorry. It says, are mutations always bad? No, they're not always bad. They can be good for the organism. So, it's like, think about coronavirus, right? So it's not always bad. It's bad for us, the host, but it can be good for the organism. It can make the organism survive better, right? So they're not always bad for the organism. Okay, now gene transfer. Let's talk about vertical and horizontal gene transfer. All right, so two ways that genetic information is passed on. Okay, so there's vertical and there's horizontal. Okay, so vertical means that we're giving it from mom to child or parent to child. I don't even know if I don't think there are any, I don't know if there are any men in this class. So, but we'll include them. We'll say parent, child. Okay. That's vertical. Okay, so that's what we normally think about. Okay, now bacteria, because they don't necessarily have, they don't have sex, right? They don't have mitosis. What they have is they have horizontal transfer. Okay, so it's going from one individual to another individual. So it would be like if you could give DNA to someone in your own generation. So it'd be like if I could give DNA to my sister. Right? I'm like, hey, you need this, right? So I'm giving it that way. So bacteria, there are some like algae and stuff like that that might do horizontal, but bacteria do horizontal. Okay, now, what did I put the wrong thing on here? Because I put on this one paper, what are the 3 types of vertical gene transfer? I'm not backwards now, right? Vertical is going this way and horizontal is going this way. So I think I messed up on the practice questions because I have what are 3 types of vertical gene transfer that occur in bacteria? And it should say horizontal. So I messed up, guys. I'm sorry. Okay, now 3 kinds of horizontal gene transfer. Do I have them listed on here? Yeah, 3 types of horizontal gene transfer. Let's put another page down here. Well, here, I have them written out. Okay, so we have transformation, transduction, and conjugation. Okay, so these are the 3 types of horizontal gene transfer. So 3 ways that bacteria can give information to other bacteria. Okay, so the first one is transformation. Okay, so transformation is basically, it's bacteria are picking up naked DNA. Okay, so they're picking up DNA that's in the environment. So this could be a plasmid, for example. Okay, so it was discovered with that experiment with the mice and the smooth and the rough pneumococcus. So I don't know if you guys need, I know I explained it on the PowerPoint, the lecture notes, but they had the mice. So there's two types of the organism. So one is rough and one is smooth, right? And they were trying to figure out which one is gonna, they're trying to understand the disease. So they took, now I'm trying to remember which one has the capsule. I'm having a brain fart. The rough probably has the capsule, but now I can't remember. The one with the capsule is the one that makes them sick, right? Here, I should look it up in the book. Hold on. I don't want to tell you the wrong thing like I have it on the other. There's the dyed mice. Encapsulated. They don't say whether it's smooth or rough. All right. It says somewhere. Okay, so we're just saying encapsulated. So if they have a capsule, okay, they're going to stick more. And if they don't have the capsule, they don't stick. Okay, so basically any bacteria that have a capsule on them are more pathogenic. But the way they figured out transformation is they put the capsulated one into the mice and the mice died. They put the no capsulated one into the mice and the mice survived, so it didn't hurt them. Okay, then they took the ones with the capsule, and they heated them, and they put them in the mice, and the mice survived. And they're like, okay, so the heat is doing something to this. So then they took the heated with the capsule and the non-capsulated, and they mixed them together, right? And you would think that the mouse would survive, but the mouse died. The mouse got pneumonia and died. So like, what the heck? So they were able to figure out that when they heated this, some of the DNA got loose, and the naked DNA was picked up by the non-capsulated ones, and that's transformation. Okay. I know, if that's a question, are we supposed to explain it? No, I'm not going to make you do that. I have made people do that, but I'm not going to make you guys do that. No. Does that make you feel better or worse? Yeah, I won't make you do it, but I was just trying to I guess get you to see that it's naked DNA. And the way that they found it was kind of crazy, right? Okay, the significance of it is important though, because that's how they make genetically engineered. So that's how they're making GMOs, or nowadays they call it a bioengineered, right? So like my Coke, I'm trying to find my Coke. My Coke says, does it say somewhere that it's made with bioengineered? No, it doesn't. I thought my high fructose corn syrup was bioengineered, but that's how they do it. That's how they make a genetically modified organism. Okay, then transduction is the DNA is delivered to the bacteria by a virus. Okay, so the virus gives it to that. All right, so that is important because of, sorry, what am I got going on here? What you guys are, okay, I need to put another piece of paper in here. So transduction, and we talk about this in the virus chapter. So this is your bacteria, and there's your chromosome. Okay, and then you have a bacteriophage, say, that comes up. And so this is supposed to be your virus, right? And this is a specific kind of virus that infects bacteria. So those are called bacteriophages, right? So the bacteriophage comes up, and I should have made its DNA a different color. Okay, so its DNA is blue, and it can inject its DNA in here, and it can integrate it into the chromosome. Okay, and basically it can change the bacteria and it can make them more pathogenic. So the ones that I told you about, diphtheria was an example of that. So you have these organisms that are the Corynebacter diphtheriae. You have them in your throat all the time, but there are some that have this piece of DNA in it from a virus. So they've been infected by a phage. So they call that a prophage. Okay, so they can give DNA. And if you guys think about it, okay, another example, which maybe this will make more sense if you think about like HPV, right? So human papillomavirus, right? So it comes up, and we're going to pretend these are cervical cells, which is not very good, but pretend these are cervical cells. And the virus comes up, and these are your chromosomes or somebody's chromosomes, right? And they can insert their piece of DNA, trying to make it a different color, their piece of DNA in here, and it causes cancer to develop. Okay, so that would be another example of transduction. So it's a way of putting virus DNA in something else. Okay, and then the last one on here is conjugation. And conjugation, I just always think of it as bacteria sex. And if you think conjugal visit, you guys know what a conjugal visit is? So conjugal, I don't know if that's how you spell it, conjugal. A conjugal visit is if somebody's like is incarcerated and their wife goes to meet them and they have like a conjugal visit where they can have sex, right? So conjugation is the same idea. So, well, they're not, the bacteria aren't in prison, but here's one bacterium, here's another one, and they can form a bridge in between them, and that bridge is the sex pilus. Okay, and they can exchange information through this pilus. So that's conjugation. So those are the three types of horizontal transfer that bacteria can do. So that's how they can get different genetic information, right? Because if they're the clones of each other, they're always going to be the same. And they can't survive that way. They always have to be changing, right? So in order to survive. So the ways that they change is conjugation, because of viruses, or because of transformation. Biotech. So biotech is really just using living organisms and living products to, they always say, to help people, right? To create something that's going to help. All right, so recombinant DNA is DNA that's recombined. It's DNA from two sources that's mixed together. In medicine, right, it's used a lot to make drugs, right? So insulin, growth factors, hemophilia factors. Okay, so it's used, if you ever see a biologic, so those say it's a biologic or biological drug, okay, that's been probably made with recombinant DNA, and they've used some sort of living organism to make it, and they can make more of it compared to like producing it chemically in a lab. Or if it's a protein's hard to make in a lab or make with chemicals, with chemistry. So they use biology to do it. Oh, I have the basic process of making recombinant DNA. Yeah. That's another good short answer question, but that I'm trying to think how you guys would do that with the online test. Right. All right, let's just walk through it so you know some basics of it. Okay, so the first thing you're going to have to do is you're going to have to find the gene of interest. So what you need to find a good gene that's not broken, right? So if you're trying to make insulin, which that was one of the first products they ever made this way. So if you're trying to make insulin, you're going to take some healthy pancreas cells, right? And you're going to pull out the gene, so the DNA that codes for insulin. Okay, so you get this little piece of DNA. Okay, then you're going to put it in some sort of vector. Okay, so probably a plasmid. Okay, and we're going to make this really simple. So we're just going to stick it in there. Okay, so this is the insulin gene. Okay, and now these have to have a way for you to be able to find it. Okay, so usually we put an antibiotic resistance gene in here. Okay, now this piece of DNA is just sitting there and you can't do anything. It's just kind of useless, right? You can't do anything with it. So the next step is you're going to transform it into maybe bacteria, because bacteria are really easy to grow. Okay, so here's my bacteria. I'm going to treat this with a chemical or electricity, and I'm going to pop this into it, which so it kind of looks like this. Okay, now what happens is I have a whole bunch of bacteria, and no matter how good I am, I can't get that plasmid into every single one of these. Okay, but I don't want to have to look at all of them to see if they all have it. So if I have this antibiotic resistance gene in here, the ones that I'm going to put them on ampicillin, say I'm using ampicillin. So I'll put them on a plate with penicillin in it. So the ones that don't have the plasmid, the ampicillin is going to kill them. So the only ones that can survive are the ones that have the trait, the good thing that I'm trying to get into them. Okay, and then I can grow these up and grow like a ton of them, and I can tell them to make insulin. I give them the right environment, they start making insulin, and then you can purify the insulin and give it to people. Okay, so I guess know these three. You have to know the gene, where the gene of interest, what the gene of interest is. You have to put it into a vector, and then you have to get it into something so you can make more copies of it. And I know that page from the book is like gigantic, like, but we can simplify it down to this. Okay, now, oh, we already talked about products, right? There's everything, all sorts of everything. Okay, now PCR, I want you to know what it stands for. So polymerase chain reaction, right? The purpose of it is to take a little piece of DNA and make a whole bunch of DNA from it. Should I write that down? Yeah. So you're going to take a small piece of DNA and you make a bunch. Okay, now, why do you care, right? Why do you care? So you care because this is used for all sorts of testing, right? So it's used for forensics. Okay, so it's a way to identify somebody at a crime. It can be used for like paternity testing or heredity kind of thing. It can be used for COVID testing. It can be used for like really any kind of testing, right? So it is important in the lab. So if you take samples, it ends up being important in order to try to figure out if somebody has something or who the father is or whatever. Okay, now there is a kind of PCR, it's called reverse transcriptase. Okay, so reverse transcriptase means well, shoot. So reverse transcriptase, we're going to tie this into viruses while we're here. Okay, so reverse transcriptase is used. So, okay, you guys remember when we make proteins, we go from DNA to RNA to protein, right? So that's the way we always go. Okay, now there is a virus that makes this enzyme that's called reverse transcriptase. And reverse transcriptase goes backwards. Okay, so we can go from RNA to DNA. Okay, so reverse transcriptase is found in HIV. Okay, so that's the first place they found it. So until HIV, this was the law. Okay, we always had this DNA to RNA to protein. But then they figured out that HIV can take RNA and it can use this enzyme, reverse transcriptase, and it goes backwards. Okay, so that's called a retrovirus. So retro, right, is going back. Okay, now reverse transcriptase, PCR, is basically, we're going to take RNA and we're going to copy it to DNA, and then we're just going to do PCR on that DNA. Okay, so like COVID is an RNA virus. SARS-CoV is an RNA virus. Okay, so the best way to figure out if somebody had it was to take the RNA, make DNA from it, and then try to amplify it and see if it's there. Okay, quantitative DNA, quantitative PCRs. Yeah, don't worry about that. Just put a line through it. Okay, now, 3 main stages of PCR and like what's required to do PCR. So those are good multiple choice questions. So to do PCR, right, you have to have some DNA. Okay, you have to have this enzyme is called Taq polymerase. And Taq polymerase is what the enzyme's going to copy. It's a DNA polymerase, so it copies the DNA. Okay, so it's what's going to copy it. This Taq part is it's thermus. And I think I always mess this up. It's like a quadus or something like that. So it's the TAQ, but it comes from the hot springs in Yellowstone. So it can withstand a lot of temperature, a lot of heat, a high temperature. Okay, so DNA, Taq polymerase, you need A's, T's, C's, and G's, right? If you're going to make DNA, you need the stuff to make it with. And then you need primers. And primers are just going to start the reaction. Okay, now the way that it works is basically 3 steps. We're going to denature, which means we're going to take double-stranded DNA and we're going to break it apart. So the temperature is high, so hot. So like boil it. Okay, then we're going to anneal. Okay, so when we anneal, what happens is we take these primers So the primers are these short pieces of DNA that are going to start the reaction. So they design these to match up with the piece of DNA you want to amplify. So like if it was COVID, right, they found a sequence in this DNA that they wanted to amplify. I think I can't remember what they were using. They're probably a spike protein. Not that that matters. Okay, so the primers stick. So anneal is stick. Okay, and then the third step is synthesis. Okay, so then for synthesis, all we're going to do is we're going to build out the DNA. So if you have A, T, C, G, A, right, you're just going to, there's a C there, we're going to put a G there, and it's going to be in the tube. Whoops. Yeah, so those are the 3 steps. So 95, this one's lower. So maybe like they say between like 40 and 60. And then this one's about 70. So multiple choice questions, I won't make you write about this. All right, now, how about viruses? So are viruses living or non-living, right? They consider them non-living, right? Because the smallest unit of life is a cell, right? And these are smaller than a cell, so they don't have any organelles. So they're not considered living. So they have nucleic acid and they have protein. Okay, so that's what they have. That's it. And you should definitely know that. Okay, so they have nucleic acids and they have protein. They don't have any organelles. They don't have, that's it. Okay, this is the thing that they have to have. Okay, some of them have envelope, right? But not everything. Okay, now 2 different kinds of viruses. So they're naked and they're enveloped. Okay, so naked don't have an envelope, and the enveloped, they do have an outer membrane. Okay, so a virion is just a virus particle. And I wish I had the picture here. This part will probably be easy for you guys, but let me find, let's see if I have chapter 14, chapter 15, no, chapter 13, because I deleted it. No. Sorry, I'm trying to find. Oh no, oh no, oh no, oh no. I want to get back. There. Okay, so let me try to draw it. Okay, so let's pretend this is a virus. Okay, and it's got nucleic acid in it. Okay, so it could be DNA or RNA. Okay, the capsid is the protein that's around it. So this is the capsid. Okay, so it's protein. Okay, so that's kind of like the shell or the wrapper. Now, the capsomere are the individual subunits of the capsid. Okay, so if I had a capsid over here and I could look at the different subunits of it, right? Each of these little pieces of it, that's a capsomere. Okay, and I put all the capsomeres together and that makes the capsid. Okay, now some viruses have an envelope around them, and the envelope is plasma membrane. Whoops. Yeah, so this is supposed to be plasma membrane. Okay, now, viruses with an envelope are easier to kill. Okay, so that envelope makes them susceptible to like alcohol and soap. Okay, so when everybody was like, oh, coronavirus, it's enveloped. So everybody's like, whoo, cool. Get out your hand sanitizer or wash your hands. They're easier to get rid of. The naked ones are harder to kill, which that seems backwards, right? Seems like if you're naked, you would be easier to hurt, but it's backwards. Okay, now I'm trying to get to here. All right, 3 basic shapes of viruses. Yeah, I'm probably not going to ask you that. Don't worry about that. Although that would be easy for you guys, probably. I never asked that. Just put a line through it. Okay, now viruses aren't named the same way other living things are named. So lots of times they're named by what the DNA or the RNA looks like, or they're named by the symptoms that they cause or what they look like. The virologists don't like when you name it after like what symptoms they cause. Like they don't like monkeypox, right? They don't want something called monkeypox, but if those names stick, they're easier to remember. Okay, now a bacteriophage or phage. Okay, so this is a virus that infects bacteria. Okay, they are really easy to study because it's easy to grow bacteria. Okay, so you grow bacteria and then you can use a virus that infects them to study the viruses and try to like understand them. Okay, now how do they replicate and the 5 steps and what's occurring in each? And I'm going to try to find the picture. We'll come back to this, okay, because I want to get into the PowerPoint and put a picture up instead of trying to draw it. Okay, and we'll come back to this too, right? Acute affection, persistent infection. I think you guys are okay with that, right? So persistent, it's in there hanging out. Chronic, it's dragging on. Acute, it's sudden. Latent would be something like herpes, right? So you have flare-ups and then it hangs out, or even like shingles and chickenpox. A retrovirus. This video is perfect. The most important thing about the retrovirus is remember that's HIV, okay, and it's going to use the enzyme reverse transcriptase. That's the big thing. Okay, and HIV, it integrates into the host genome, right? So you have it for life. They haven't figured out a way to get it out of people's body yet. Okay, so let's see. Let's finish this page and then we'll go back. Okay, now what's required to culture a virus? Okay, so you can't grow a virus in a petri dish. So you can't grow it on agar. You have to have a living cell. So you have to have a cell, you have to have a host. Okay, so you can't grow it on agar. It doesn't work. So that's like a gimme question. I'm going to ask you that. You guys are going to get it right. Okay, so you have to have a living cell. You have to have a host. Eggs in the past, right? And they still do grow it in eggs. So they'll grow the flu virus in eggs in order to come up with a whole bunch of the virus in order to make the vaccine. Okay, so eggs, they just get a whole bunch of eggs and like robot arms inoculate them and then they harvest it. They also do it in cell culture, so they can grow cells in like kind of like petri dish, some a little more sophisticated than that, but they can grow the cells in culture and then they can infect the cells. Okay, but you have to have a living host, okay, because they're obligate intracellular parasites. some viruses cause cancer because where they integrate into the host genome. Okay, so if they integrate into the host genome and they interfere with the gene that has to do with mitosis, okay, they're going to cause cancer, or they could potentially cause cancer. Okay, so some of them that are associated, you guys remember HPV, right? HIV used to, but that's less common. Hepatitis, right? Liver cancer, those are the big ones. There's some other ones, but those are the big ones. Oh, and I have this silly question about plant viruses. Okay, so like plants don't walk around and cough on each other, I think, but they're transmitted by the plants have to have a wound. Okay, so the plants have to be damaged. So like if an insect comes up and it starts eating the plant, the virus can get in through that wound. And this is neither here nor there, but it's kind of cool. All right, now let's go back to this stuff, how the bacteriophages replicate. Are you guys still with me? Are you still there? All right, let me try to find, I'm gonna go back out to to find a picture. Let's see if I can find a picture here. I think I have one here. No. Yeah, I have this picture. Okay, so download. I don't know what I would do without my iPad. And the best part of it is the Apple Pencil. All right, now you guys help me. I have to find the one that says, like, they don't come in a regular order. The one that I just did. Where is it? Oh, shoot. The downloads. I don't see it. You guys probably see it. And then chapter 2. Oh, crap. All right, let me try again. It should be here though. Download. Good notes. New. I don't even know what the file was called. I'm getting tired. What's it called? Review, is that really what it's called? Exam 2, right. Okay, so let's do this. Part. Exam. To review. Oh, this, look how many times I have it there. All right. Oh, this is lovely. It messed it up. I would just not even be able to see. Oh, there it is. Cool. Okay, so let's go through this. I wish you guys I could share my screen and one of you could write on it. Okay, so let's go through this. And this picture is from your book, and it's on page 13. Well, no, it's figure 13.5. That helps. Okay, now attachment. Okay, so that's the first stage. And even though this is bacteriophage, pretty much every virus has to work this way. Okay, so they have to be able to attach to the cell. And that's basically all that's happening. And if you think about coronavirus, okay, they have spike proteins on them, right? And they attach to the ACE receptors on our cells. So these spike things, that's what's going to attach to a receptor. So you can think of a receptor like a little cup, right? I always think of it like, do you remember those sticky things you get at the Dollar Tree that you throw the tennis ball and it sticks? Okay, so the sticky thing is the receptor. Okay, and the tennis ball might have spikes on it that are going to attach. Okay, so attachments first. Okay, then the virus is going to insert its genome. So this is genome entry. Okay, so totally being violated, right? The virus comes up, it almost has like I always think of this as like the War of the Worlds or Chicken Little. Okay, so the alien, which is the virus, attaches, it lands, attaches, and then it injects its DNA or its RNA, its DNA into the host. Okay, then what it's going to do is it's going to basically hijack, okay, the host cell, and it's going to tell the host cell to make the virus. All right, so this is my description of it. Technically, it's called synthesis. Okay, so synthesis is when the host cell gets pretty much hijacked. Okay, and it starts saying, okay, the virus is saying, I want to make more virus. In order to succeed, I need more virus. So I need you, bacterial cell, to make my DNA and to make my proteins. So I can make more virus particles so I can keep spreading. Okay, then the next part is pretty easy, and that's assembly, right? So assembly just means we're putting the parts together. Okay, we're making new viruses. Okay, so we make new viruses, and then this is lytic. So lytic, this one is breaking apart, right? So this is release. Okay, and what's going to happen then is the virus can go out and it can spread, and it can go wherever. Okay, now sometimes, okay, so this is considered lytic. So lytic is like lysing, which this is a frustrating naming thing to me because the names are too similar, but I didn't do it. Okay, so lytic, this one's lysing. So lysing means it's breaking apart, it's bursting. Okay, now lysogenic means it gets in here, which now this is bad picture. Okay, it gets in here, and what happens is the virus becomes part of the host. Okay, so it just integrates into there and it forms, this is called a prophage. All right, so then this can divide and it keeps the prophage. So this is what they call the lysogenic infection, which I know there's probably some very good reason why the names are so similar, but I wish this was named something different. Okay, so we can go into this lysogenic and it can hang out like that for a while, right? And then it can like kick it back into this gear. Now it's interesting because bacteriophages they're everywhere, right? If you watched any videos about them, they're totally everywhere. You guys could go outside and you could find them in dirt, right? They use them in place of antibiotics sometimes. So if you have an antibiotic resistant infection, right? So you have this bacteria that are causing a problem in a human, right? And you can't get rid of it. The antibiotics aren't working. They have this collection of phages, of bacteriophages that they bring in, that they could bring in, and the phage will kill the bacteria. Okay, now, it's not been approved in the United States, right? It's like an emergency use only thing. It's been used in other countries. So Georgia, which was not Georgia state, but Georgia used to be part of the Soviet Union. Okay, so they use them there, right? They use them other places, but we don't use them in the US. One reason could be because of the possibility of developing these prophages, and that would make this thing, could potentially make this bacteria more pathogenic. All right, now I'm squirreling. Okay, let's go back here. All right, now, oh, I put this in here. I get delirious. The first one discovered, do you guys remember what it is? This is tobacco mosaic virus. This is the first one. That's a good, you get the, I'll put that on there since you guys were here and you'll get that right. All right, now how do you feel about the immune system? I don't know if you guys need me to go over that again. Usually micro students, you're all fine with that because you have it in A&P. Are you okay with it? And like, I really pared it down. So if you look at the videos that I made and the PowerPoint, you might be fine. Yeah, what part, Michaela? Is there a specific part? Yeah, some of you feel more confident, right? Okay, well, let's just hit. Okay, so remember, innate and adaptive. Okay, so innate, remember that is the one you're born with. Right, so that's the one you're born with. And I honestly, on these immune questions, it would be really easy to make it really, really hard, but I don't try to do that. I just try to keep it pretty straightforward. So hopefully it's, you guys feel the same way. I think it'll be fine. Okay, so innate is what you're born with. So make sure you can give examples of innate immune system, right? So like your skin, your mucous membranes, inflammation, so that kind of thing. Okay, then adaptive is what you develop over time, and it's way more specific. Okay, and this is what we make memory to. Okay, so now an antigen is anything that causes an immune response. Okay, so it can be, I don't know, there's lots of pollen now, or it could be a virus, it could be part of bacteria, right? So an antigen is anything that causes an immune response. And then an antibody is what our body makes in response to an antigen. And that's part of our adaptive immune system. Right, so that's specific. They're proteins. They're Y-shaped. Okay, now I just wrote this out here. What are the types of innate defenses? So the first line defense is going to prevent entry. So that's like your skin, right? Mucous membranes, your tears, saliva, that kind of thing. This is the way our book breaks it down, and I know the anatomy book doesn't break it down exactly the same way, but they're pretty similar. Okay, now we also have innate defenses that are going to detect invaders in general. Okay, so they call these pattern recognition receptors, and I don't remember this in the anatomy book at all. Okay, but they're going to detect like, oh, there's bacteria in here, or oh, there's a virus. They're not specific to whatever the virus is, or they could say the tissue is damaged. We need to do something. We need to rally the forces. Okay, and then the complement system is this, it's actually like a complex system of proteins and like a cascade, a biochemical pathway. But I just look at what I put in there. I made it like super simple, right? So the complement system, I can pull up a picture, but it's like you want to look at opsonization. So it's going to enhance phagocytosis. It's going to communicate and so inflammation, opsonization. So we're going to enhance phagocytosis. We're going to cause blood cells to rush to the area, and we're going to lyse cells. Okay, then the innate defense, the other thing that's going to do is it's going to eliminate the threat, right? So interferon, right? So do you remember interferon? I want you to always think of virus. Okay, so if interferon is released, it's in response to a cell that's infected with a virus. Okay, then phagocytosis, the complement system again, inflammation, and then fever is how we're going to get rid of it. What are physical barriers to entry? We kind of talked about that. Antimicrobial substances that are going to prevent entry. Let's see, so tears and saliva, right? They have lysozyme in them, which I'm spelling wrong. I know, I guess I'm running out of steam, you guys. Let's see, I'm going back to the lysing enzyme. We'll have peroxidases. There's some antibodies in there. I know, I'm just like, I probably won't. The big one that I always focus on is lysozyme, and that it's in your tears and your saliva, right? Okay, now, what did I do? This looks ugly. What are the types of white blood cells? What do they do? When would they be elevated? Table 14.1. Why does this look so bad? Maybe it was in a chart. Probably. It was probably in a chart. And when I imported it, the chart got messed up. Okay, so let's do this. So granulocytes, you guys remember. Yeah, yours looks like a chart. Mine got butchered when I downloaded it and imported it. Dropped my Coke cap. Okay, so granulocytes, these are white blood cells that have grains in the cytoplasm. Right, so I just remember they have grains. And the grains are granules, and the granules are going to actually help the white blood cells kill the bad guys. Okay, so now the granulocytes, we're going to break down. I always say, remember Ben. So Ben loves his granny. Okay, so basophils, eosinophils, and neutrophils. Okay, so now I'm always trying to get you to think about like what makes something different from the other things. Okay, so basophils are the only ones of these that they don't do phagocytosis. So they don't do phagocytosis. Okay, so they're not going to engulf anybody. They don't. Okay, but what they do is they release histamine. Okay, so they release histamine. So that's what makes them unique. Okay, now eosinophils, you usually find if someone has allergies, if there's an allergic response, and you can find them around parasites. So if somebody has like a tapeworm or something, they're going to surround them. Okay, and then neutrophils, they are the ones that occur in the highest number. So they're most likely to be the first ones to get to the scene. Yeah, so that kind of makes sense, I hope. Okay, so now mononuclear phagocytes are the monocytes, the macrophages, and the dendritic cells. Okay, so let me go over here. Okay, so we're going to have monocytes, macrophages, which I'll write it out, and dendritic cells. Okay, so monocytes are the biggest white blood cell. Okay, so they're huge. And they call them mononuclear phagocytes. So their nucleus looks just more like one piece compared to the other ones. Okay, so these are gonna do phagocytosis. That's their big thing. Okay, now monocytes are called monocytes when they're in the blood. Okay, but they can leave the blood and they can enter the tissue. Okay, so if they enter the tissue, they're called macrophages. Okay, so this is when they're in the tissue. So basically the same thing, just a different location. Okay, so they enter the tissue. If you had anatomy, there are a bunch of macrophages that are, they're kind of like permanent. So they're always found in the same locations, where there are other ones that are transient. And the transient ones are going to move around throughout the body. Okay, then dendritic cells, okay, they're in the tissue and they're looking for things that aren't self, and they're just trying to get rid of it, and they're going to let everybody else know that it was there. So that's like really simple. But I would know, like, these are good multiple choice questions, right? To say what following aren't matched correctly. Which of the following does not phagocytize, which occurs in the highest number, which will be present if there's parasite. Okay, now lymphocytes, okay, are subcategorized into T cells and B cells, right? So these are part of your adaptive immune system. Okay, and the main things I want you to get out of this, okay, so the T cells are responsible for humoral immunity. No, you guys, somebody should have yelled at me. Well, you can't yell at me because you don't have your sound. But okay, T cells. Okay, B cells are humoral. Humoral immunity. So this is antibodies. So remember B and B, right? And then T cells are cellular immunity. Okay, and then we talk about the different kinds of T cells. Do I have that on here or no? Yeah, a little bit. Okay, so we'll talk about it. And then NK or natural killer, which they're just going to destroy things that are bad. Okay, now our book talks about cell communication, right? So they're saying receptors on our cells are the eyes and the ears. Okay, and then in order to talk, right, we have to have cytokines. So cytokines are the chemicals. So there are things called chemokines, which I'm just going like really general with it. You don't have to worry about it too much. Okay, colony stimulating factors, which are going to cause the cell, like this is going to cause the cells to divide, multiply, increase. Interferon, okay, which you're always going to think of virus. Okay, interleukins, which those are kind of like a checks and balances system. And then tumor necrosis factor is a way to take out tumors. I know, I'm just like, some of this is kind of redundant. So pattern recognition receptors, they're going to look for something that looks like it's bad, like it doesn't belong. And you guys can look in the notes, there's a interferon. There is, I know I talked about it, and there's a really good picture. So watch that. Complement system, just know in general, there's not a lot that I'm not making you, there is a lot, but I don't require you to know all the details about it. And then you guys know what phagocytosis is and inflammation. So the goal of inflammation is to really get more red blood cells, or not red, white blood cells to the site. Right, so it's going to make it hot. There's going to be more blood there. Might make it hurt, so you don't move. And there's swelling, but the whole goal is to get white blood cells to where it is. Oh, I have, what's a pyrogen? What's a pyrogen? So if you think pyro, like pyromaniac, so pyrogen causes a fever. So like ibuprofen is an antipyretic. Then over here, histamine. So histamine's coming from basophils, and it makes capillaries leaky. Okay, so it makes them leaky, so it's going to put more water into the system. You think about, like, if you're allergic to something and you're having a whole bunch of histamine being released, right? Your nose is running, your eyes are watering. Okay, the benefits of a fever, it increases the body temperature, and that actually will speed up processes in your body, and it will inhibit the growth of the bacteria. Okay, then adaptive is what we develop over time, right? So we develop it after we're exposed to antigens. So it's specific. So memory is how we actually like remember what we've been exposed to. And that's why a vaccine will work, right? Because you develop memory to a certain antigen, so then you can respond to it faster. Immunological tolerance is the idea that your T cells and B cells are made so that they don't attack your body. And it's kind of complex, but I just want you to know in general what it is. Okay, and then T cells, right? So T cells are cellular immunity. And you guys can look at the notes. Okay, they mature in the thymus. Okay, they have two different kinds of receptors. So there's CD4 receptors and CD8 receptors. So CD4 and CD8, which are which? Does anybody remember? So I'm going to mess it up because I'm tired. Which one has the, I've only ever said this like a gazillion times. So a cytotoxic T and helper T, which one's got which receptor? Who remembers? Does anybody remember? I'm looking in the book and I don't remember which one's which. That's not good. I don't even see it in our book. I know it's got to be in there. I guess I won't ask you that if I can't find it in our book, but I can't remember. I think CD4s are the general. Those are the helper. But let's just put the T helper cells. So those are the ones like the, yeah, right. Thank you. I was right. But I really don't see it in our book. How can that even be? But anyhow, so T helper, these are like the general of the army, right? And then the CD8 are the cytotoxic T cells. Thank you, Lexi. I'm getting tired. So you guys can go back and watch the video, right? And you know the difference between cellular and humoral. Make sure you know that. The antibodies, so IgM, IgG, and IgA. So there, I just had put I put the 3 types on here for you. So IgG is the one that is in the highest quantity, right? It can cross the placenta. I know, I'm getting tired. I'm not making any sense. Okay, and the natural killer cells are cells, they're going to go out and attack. And even though we have them under this adaptive immunity, they're part of the innate immunity. All right. Questions? Are you all still with me? Yes. If you have questions after you start studying, you're just going to email me. And if it's something big that I missed, I'll post it on the announcements on the class. Does that make sense? I know, I hope it helps. Another thing you can do, like, does this book have, like at the ends of the chapters, although I don't cover everything, but at the ends of the chapters, there are review questions. Yes, I'll post the key to the practice questions. I'll do that in the morning. I'll fix it because there's a mistake, but I will fix it and post it tomorrow. I just have to write them down and scan it. At the ends of the chapters, there are multiple choice questions, and they're really similar to the questions in the test bank. Hint, hint, hint. The other thing that I've seen is that when people do the like the Learn Smart, which I don't know if that's what it's called anymore, the Learn Smart stuff, that will help too. And I'll post this, I'll try to get this. Sometimes the Zoom stuff takes a while to render. So I will, if I don't get it posted this evening, I'll get it posted first thing in the morning. Like they don't always, in my experience, they don't always get into the, they, like Zoom takes them and it makes them, I think, MP4 or something. And then then I can put that on YouTube. So yes, I'll try to get it done if it renders in a reasonable amount of time. Right? Anything else? Did you have a good spring break? Yeah, use the chapters questions from the ends of the chapters. Some of them you'll be like, what? But so if you are like, what? Because we didn't talk about it, then just ignore them. But others make really good sense. And they're very similar to the stuff in the test bank. And some of them like opportunistic pathogens are least likely to affect which of the following groups. And like, AIDS patients, drug addicts, cancer patients, college students. They're at the ends of the chapters in the book. So you should be able to, I can post links to them if you want. I guess I can probably post link to them, but at the ends of the chapters, there's some short answer questions. And some of them might be useful and the others might not be. Oh, I'm looking at the wrong chapter. I read questions from the wrong chapter. I'm sorry. I read it from 16, which I made the video for. Does that mean we can use our notes for, no, you can't use them. You can't use your notes to take the exam, but I wanted you to be able to focus in on what to study because there's way more stuff in chapter 7, 8, and 9. Yeah, I know, sorry, I meant to study. The next lab test is open book. Yep. Yep. Yeah, right. I don't know if you'll thank me. You might be like going, what?
Can I ask a quick question about lab?
Yeah.
For the dropped labs, do you plan on dropping just the Connect or HOL, the lowest two, in a whole of labs, or just the Connect?
Right. I just dropped the Connect so far. I don't know what to do with the HOL because I want everybody to be doing them. But I'm thinking if you've done all of them, that I might drop the lowest one. Does that make any sense?
It does. I wasn't sure, since we're guinea pigs with it, if there was going to be like some consideration about like including those in the drop, because some of them were a little rough to understand.
Yeah, yeah, I know. Right.
Yeah, that would be great if you would consider
Okay.
those as
If you
well.
do them all, I'm more right.
Yeah.
But some people aren't doing them, but they're not here. So
Oh no, I'm doing all of them.
yeah, right.
Yeah, I was the one that
Yeah.
emailed you earlier because mine didn't, like, my disc didn't grow right and my agar.
I know. That's not your fault.
Yeah,
I know.
it was a mess.
It's
Thank
okay.
you.
Yeah, we're getting there. It's all right. All right. Well, then have a good night. And if you have a question, send me email. Okay, thanks. Thanks for coming. This might be like a record that I have 11 people here. Woohoo.
Thank you.
Yeah, I don't know if is that good or bad. No. Bye, guys. I don't know if you can see me. You probably can't see me anymore there. Can you see me?
We can, yeah.
Oh no, have you seen me the whole time?
The whole time, yeah.
Great. So did I pick my nose or anything?
No, you were good.
I was like messing with my hair and trying to drink my Coke.
No,
All
you
right.
were
And my
thank
screen
you.
is, something's wrong with my camera. It's black and white.
It is black and white, but yeah, we could see the whole time, but you're good. Thank you. We appreciate it.
I
know
didn't know that you could see me the whole time. Oh boy. All right. Bye.
Bye.
Bye.